pairs of double-stranded hairpin available for XPD to unwind. So in these experiments, we hold the DNA at tension and incubate in XPD until a molecule of XPD binds. Then, upon exposure to ATP, XPD begins unwinding the hairpin, and since we're at constant force, the total extension and the distance between our two traps changes. And by tracking that change in extension, we can calculate the number of base pairs that XPD unwinds over time. So that's what all the data I'm going to show you with this is mostly, this base pairs per time that XPD unwinds. So I've hinted that we incubate in XPD and it's exposed to ATP, but how do we control this exposure to ATP, the exposure to XPD, loading these proteins? So we accomplish that with a laminar flow cell. This is a picture of the actual flow cell. And if we zoom into the experimental area, there are 3 streams that meet. So this is food dye in the blue food dye in the top stream, red food dye in the middle stream, and nothing, or red on the bottom and nothing in the middle. And you can see a really clear distinction between each stream, which allows us to have different buffer conditions and move from stream to stream with our DNA construct. So in my experiments, I have XPD in the bottom stream and ATP in the top stream. I hold the DNA at tension first in the center blank channel, and then move into XPD to incubate for 30 to 60 seconds till a molecule binds.
So
Yes.
How do you know when the binding occurs?
You don't.
You don't.
You just wait a while, hope for the best, move, and if you see unwinding, then something was there, and if you don't, then you're unlucky. It's about 30% of the time I see binding and unwinding.
Okay.
So yes, and since our loading site is only big enough for a single XPD, we know there's only one there, and it doesn't start unwinding until we move into ATP. So for demonstrative purposes, I'll show you that whole thing again, but this time with the data that we take. And I've loaded a fluorescent dye into the central channel. So that's just for illustrative purposes. I'm not going to show you any fluorescence data after this. Just when this dye intensity is high, it means we're in the middle, and when it's low, it means we're not anymore. So we begin in the center with this high intensity. No base pairs open, our hairpin is closed at high force. I then lower the force and incubate in XPD for about a minute. And then I've covered up the number of base pairs open because at these low forces, the calculation is inaccurate. The force is so low, it should be closed. Then moving into ATP, I increase the force, and as soon as XPD is exposed to ATP, it begins to unwind, and we see this activity.
Last question.
Yeah.
So how fast do you move it across the chamber?
This, from the bottom to the top, takes about 10 seconds, 10 to 15.
But it's like very sharp in your point.
Yeah. Oh,
it's like,
this bit.
It's like instantaneous.
Right. So the changing of the buffer conditions is instantaneous. Moving from here, where we have XPD, to where we have ATP is, the duration of this green chunk, which is about 10 seconds. So yes, changing of conditions is very fast. And, if we could shrink this, previous experiments have done XPD and ATP in completely adjacent streams so that you just instantly move from XPD to ATP. Out of an abundance of caution, particularly with using RPA, which has melting, I've added this blank channel just to know that they're definitely separated.
And the force, you also load instantaneously?
Yes. So it's a force feedback mechanism.
Or this is what you think you do? Like it's what
this
you
is
said
actual data. Yeah, so this is the actual force, 12 piconewtons. I turn off force feedback and it's low, and when I turn it on again, it's instantly 12, more or less.
It's a little bit.
Well, not much, but yeah.
Seconds. Okay.
Yeah, and increasing the force I do in the center, so by the time it enters ATP, it's at constant force and ready to go. So taking a closer look at the unwinding data that I get. So you see XPD unwinds in these, what I call bursts with limited processivity. So one burst consists of XPD moving forward for some significant duration and then back stepping or back sliding, often back to the start of the hairpin. Not all the time back to the start, but often back to the start. And these bursts have limited processivity, which is the number of bursts or the number of base pairs XPD unwinds in one attempt. So you can see that, each of these bursts only unwinds somewhere around 15 base pairs, and it has available 89 base pairs to unwind. So that's not that much.
Why does it go back?
That's a really good question. So I haven't studied the backward motion in detail. I think it's very worthwhile. We know from previous experiments, and he in my lab previously did more kinetic models of the forward stepping and backward stepping rates. XPD is just very prone to back stepping. You'll also see in the next few traces that XPD, I believe, strand switches. So I don't know the exact conditions that make that more likely or less likely, though that's definitely an interesting question.
But if you lower your force, would it backstep more readily or less readily?
If you lower the force, I'm not sure about backstepping. Certainly the forward stepping decreases at lower forces so that you see just lower processivity the lower you make the force. It just can't go as far. It needs that extra applied force to help it. The backstepping, I would speculate if you increase the force, you're going to increase the overall processivity and perhaps see less backstepping. But I haven't done too many experiments with the changing force. All of these experiments are constant 12 piconewton force, and I vary RPA conditions.
12 piconewton is not too far from 15 when you will just unzip the DNA, right?
Yeah, it's not too far. It's a pretty weak helicase. So XPD unwinds in these low processivity bursts. So what happens in the presence of RPA2? And to do these experiments, it's the same thing, but now I've mixed RPA2 into the ATP stream. So incubate an XPD, pick up a single XPD, and move into ATP plus RPA. And so now as XPD unwinds, RPA2 is in solution to bind and interact as it will. So in these experiments, I use RPA2 concentrations between 0 and 50 nanomolar, and as I was saying before, always 12 piconewton force, and always 500 micromolar ATP. And you can see as we increase the concentration of RPA, more and more XPD is able to unwind more and more of the hairpin. So again, processivity defined. This means the number of base pairs that XPD was able to unwind in one attempt. So each of these traces, again, is one single XPD. And you can see that processivity varies throughout XPD's unwinding. In this case, XPD starts with low processivity, then goes farther on its next attempt, low again, and so forth. And I'll say, since you mentioned the backstepping, backsliding, this is an example of a trace that, or a burst that I would say is strand switching, XPD moving forward, switching strands, and going backward. These cases where you sort of see a flat top on them is XPD moving all the way through the entire hairpin, getting to the top 89 base pairs where, that's it. It stops at 90 or 89. And then continuing through to the other side, and the hairpin re-anneals behind it. So yeah, we have this increasing overall processivity.
So it can go through a single strand DNA.
Yes, translocate.
Why doesn't go and unwind all your tethers?
Because I have an A basic site at the other end. So do I have a good?
That's good.
Okay, yeah. There's an A basic site that can't go through.
It was in your hairpin.
Yeah.
You didn't know the schematic you had.
Yeah, it was. I glossed over the A basic site. Yeah, so the overall processivity increases with RPA2. So I divide these traces into independent unwinding bursts and analyze them as independent attempts at unwinding the entire hairpin. And so this plot shows each point is one burst and the processivity, the maximum number of base pairs XPD unwound during that burst. So you can see the average, these white dots, is increasing with RPA concentration. You can also see the distribution. There's this large population of low processivity unwinding with this sort of long tail of higher processivity that is even present without RPA there. And as we increase the concentration, we see more and more bursts fall into this tail. So I created a threshold of 25 base pairs to just distinguish between this low processivity population and this high processivity tail, and maybe give us a little more insight into the differences in unwinding. And the fraction, perhaps obviously from this figure, is the fraction of highly processive bursts. The fraction in this high processivity tail is increasing with RPA2 concentration. So we've established that RPA is enhancing XPD processivity. Now I'm going to talk about some proposed mechanisms for how RPA is enhancing XPD processivity. So the first one is this sequestration model, where RPA binds behind XPD and prevents it from backstepping and backsliding. So if XPD can't step backwards, it's sort of forced to go forward. There's also a duplex melting model in which RPA2 binds near the junction and melts the DNA ahead of XPD, making it easier for XPD to move forward and unwind. And then finally, a complex formation model where XPD and RPA2 directly interact, and the two of them together are more processive than XPD alone. So I'm going to start with this sequestration model and talk to you about some of the evidence. So under the sequestration model, we're decreasing the rate of backstepping and backsliding. So if XPD is not backstepping and backsliding so much, then we should expect to see bursts of longer duration. So we've sort of overall lowered, like this is so hard to say still, but we're not backstepping and backsliding as much. So we'd expect these backsteps and backslides to happen after a longer time, increasing the total duration, time duration of each burst. So if RPA2 is affecting these duration of unwinding bursts, then it could be indicative of this sequestration model. So this is a plot of low processivity bursts in gray and their average duration, and high processivity bursts in white, their duration, and the average of all bursts at each concentration. So you can see overall, the burst duration is increasing, but this is entirely due to these highly processive bursts, which are overall much longer in duration than these low processivity bursts. For the high processivity and low processivity separately, the trend is essentially flat. RPA2 doesn't affect their duration at all. So this evidence seems to rule out the sequestration model. So next I'll talk to you about the duplex melting model, where RPA2 melts DNA ahead of the duplex. So on its own, which I have not told you yet, but here it is, RPA2 is able to melt or break the bonds, firmly break the bonds of DNA on its own. So in these experiments, I have just RPA, no ATP, no XPD, and I hold the DNA construct at tension in RPA and allow it to bind and unbind. And we see this sort of characteristic blip signal when in the presence of RPA. So these melting events are about 5 to 10 base pairs in magnitude and very, very short, about 20 milliseconds. So RPA2 can open the hairpin. And we even see this during XPD unwinding in RPA, in the presence of RPA. These sort of same characteristic blip events, short, high, about 5 to 10 base pairs melted, similar to what we see RPA2 alone doing. So we wondered, well, we can see these melting events. Are these melting events helping XPD to unwind? So we use a step fitting algorithm to fit this unwinding data and select RPA2-like melting events out of the data. So the proportion of these events increases with RPA2 concentration, indicating this isn't just a fluke or noise. There's some false positives, like at 0 nanomolars, but the majority do correspond to RPA, an effect of RPA. So yes, this indicates RPA2 is able to interact with that duplex junction while XPD is unwinding. But does this help XPD to unwind, is the question. So to visualize this effect, I picked out each blip during unwinding, and I aligned the before and after. So I aligned both the before and after to set the initial DNA unwound to be 0. And then I aligned the re-annealing of the hairpin in time in the after. So, and then I took a heat map of the results. So if there's a majority of events where we have more DNA unwound than we started with, that would indicate RPA2 is helping XPD to move forward. If we end up with more data points below this line, that indicates RPA2 is hindering XPD somehow or sending it backwards. And if there's no difference, then there's essentially no effect of RPA2. So here's the heat map of these before and after. So this is before and after a melting event. And the white line is 0 or no change. And there's perhaps a slight peak above 0 here. But it's no different from selecting a random location and looking at a time 50 milliseconds later. So RPA2 doesn't seem to help XPD unwind anymore, or at least these melting events aren't helping XPD to unwind any more than it already is. So to me, that indicates the duplex model is out. Yes, RPA2 is melting, but it's not helping XPD move forward. So now we'll look at the conformation or complex formation model of XPD and RPA. And to do this, I do a slightly different variation on the same experiment. So now we have XPD and RPA2 mixed together in this bottom channel and ATP in the top. So when I incubate, XPD will bind, and if a complex is forming, the XPD-RPA2 complex will bind. And then moving into ATP, we'll bring the complex into ATP, and we'll see that complex unwind, if it exists. So increasing the RPA2 in the bottom channel should also increase the likelihood of forming a complex. And thus, if a complex is forming, we should see an increase in processive activity as we increase RPA2 concentration. However, even at very high, 200 nanomolar RPA2, we don't see any change in the highly processive fraction of bursts, indicating that there is no complex forming. We're just seeing XPD binding even at these high concentrations. So this rules out a long-lived complex, but there is a caveat. Going back to your earlier question, so there's about a 10 seconds of motion moving from the XPD channel to the ATP channel. So if this is a very transient complex that falls apart in less than 10 seconds, we will not see it in this data. But this does rule out a tight binding, long-lived complex.
But this is a very indirect way of quantifying complex formation.
Is it?
Well, I mean, you're looking at the action of helicase,
Yes.
whereas you could do something like FRET or,
So,
and just see directly whether it forms a complex or not.
in my thesis, I haven't gone into it here, but we do FRET, similar experiments using FRET. FRET just tells you co-localization.
Right.
It doesn't necessarily tell you RPA is stuck to XPD.
Well,
So-
what if it's not there? It's definitely not stuck.
True. So my dipping experiments using FRET, I did not see any FRET signal
So it's not there.
indicating RPA. However, I rarely saw any FRET signal at all. So it was not a successful experiment. I will say this is supported. Our collaborators did immuno pull-down assays where they had an antibody for, I think, XPD, and they mixed XPD and RPA and pulled down the XPD. If there was a tight binding construct or complex, then they would expect to see RPA in that sample too. But they didn't see that forming in solution.
Well, then this, I don't know why you did this then.
So
It doesn't bind,
this was a model proposed by our collaborators, and they thought it could be something DNA mediated,
okay.
or which again, when you throw DNA in the mix, you can't really tell the difference between both being bound to DNA versus being in a complex on DNA. So
I can add a thing about the FRET experiments. At those concentrations, it's really hard to do because you get a background from the free protein in solution. So Barbara did not include in her talk, but she spent many months actually trying to do that experiment.
I believe that.
It's a very difficult experiment that did not end up being successful. So this is the best that we can do.
So yes, this indicates that the complex likely is not forming, with that caveat of it being a long-lived stable complex. But again, a transient complex, that interacts for less than 10 seconds is still possible, is not ruled out by our data. So after ruling out every possible mechanism, we sort of go back to the drawing board. So we've taking a second look at our XPD unwinding data. Clearly there is an effect of RPA2. We see this highly processive fraction increasing in the presence of RPA. Oh, and just to be clear, I'm moving away from those complex formation experiments. This is the old data. XPD alone is in the bottom channel. There's unwinding with RPA2 mixed with ATP. So there is an effect, and we do see more unwinding bursts in the tail of this distribution. So what is the difference between low processivity unwinding and high processivity unwinding? So I took just the unwinding part of each of these bursts, and that should have been animated. Align it to 0 time, just to look at sort of the overall picture of what XPD unwinding looks like. And you see a lot of sort of similar features, even as we increase RPA2. So here we have, these long stalling behavior at 10 base pairs is pretty consistently seen at every concentration, including very high RPA2. And if I look at the average unwinding burst for low processivity and high processivity at each concentration, you see remarkable similarity at all concentrations of RPA2. These low processivity bursts tend to just stall out around 10 base pairs, and the high processivity bursts just keep going, and at the same velocity, the same rate, whatever the concentration of RPA2 is. So the fact that RPA2 doesn't seem to be changing these features of XPD unwinding indicates that they're features inherent to XPD itself. So we propose a 2-state model for XPD processivity, where XPD has 2 inherent states, one low processivity and one high processivity. And the presence of RPA2 increases the likelihood that XPD is in this highly processive state. And this is sort of mechanism agnostic. There's a broad range of possibilities for the physical underpinnings of what caused the low processivity versus high processivity. But we can use this model to describe our data very well. So we created this kinetic model of XPD processivity. So there are sort of 2 + 1 states, the low processivity state, the high processivity state of XPD, and an off state where XPD completely dissociates. And again, adding RPA would increase this rate into the high processivity state. So we can't directly track this low processivity and high processivity state. They're, inherent states to XPD that we believe exist based on the processivity signal that we get. But we can see the effect, which is crossing this 25 base pair threshold. So if we look at a first passage time for how long it takes a molecule of XPD to reach 25 base pairs, that's going to vary between molecules and also with the concentration of RPA. We measure that time from the start of unwinding. So a first passage time model would look like this. We disallow transitions out of the high processivity state, and I will spare you all the math, but we can calculate the probability of XPD being in this crossing into this high processivity state by time t. And we can also calculate this directly from our data, the percentage of molecules that have crossed our processivity threshold at each time point. So this is the raw data for the proportion of molecules that have crossed our threshold by time t. And as we increase the RPA concentration, you can see this increases faster. And fitting it to our model, I did a global fit with the same value for K off, the same likelihood of dissociation, but I allowed the rate of processivity to vary with concentration. And found that rate of entering the processive state increases linearly with RPA2 concentration, which implies for biologists anyway, implies a direct interaction between XPD and RPA, direct one-to-one interaction. So just to remind you, this two-state model, we have a low processivity state, a high processivity state, and RPA2 increases the likelihood of high over low. But again, there's room for broad interpretation of the physical underpinnings of these states. And for future work, we are going to need to look into what is physically causing these two different states. So I'm going to tell you a couple ideas about what that could be. So one is this transient direct interaction that we haven't been able to rule out. So XPD is known, and our collaborators demonstrated this in some nice fluorescence experiments, to have two conformational states, one open and one closed. So we still don't know exactly what these two conformational states do, but wouldn't it be cool if one was a low processivity unwinding state and the other was a high processivity unwinding state? And perhaps a direct interaction of XPD with RPA2 could influence XPD to be in one conformational state over the other. There is evidence of direct interaction of XPD-like helicases with RPA or human RPA, as well as human XPD interacting with its neighbors in transcription factor 2H. So this is a tempting model, and it would be great to be able to do experiments that give further insight into what these two arch domain states, their effect on unwinding. So another possibility for these two states is a secondary binding site on XPD for single-stranded DNA that binds to the opposite strand as XPD unwinds. So this idea is that this secondary strand, or displaced strand as it's called, binds to XPD, and in our experiments, maybe that binding could be hindering XPD's forward progress, since we're applying force, and if it's a strong enough bond, that would pull XPD back. RPA2 is a single-stranded DNA binding protein, so if RPA binds to this displaced strand, it would prevent it from binding to XPD. So this can be tested using a modified DNA substrate that was done with REQ helicase and another group they had a similar hairpin assay and found that display strand binding was preventing RecQ from unwinding processively, causing long stalling behavior. And switching to a different complex construct geometry, still allowed for display strand binding, but that binding no longer hindered XPD or hindered the protein's forward progress. So if we can use a similar construct with our XPD molecule, we could see how that changes XPD unwinding, if at all. And with that, if there's no questions, I want to thank my committee, my advisor, all the members of Shenmore Lab, and funding, and all of you for listening to me.
Are there any questions?
TJ has a question.
Any questions, TJ?
Yes, I have a question about the model.
Yes. Want the math or just the basics?
So, do you have information about the lifetime of the highly processive state?
Sorry, we just got some noises. So we cannot probe that directly. Jan and I are currently working on a slightly modified model that involves a detection step that gives a bit more insight into that lifetime. We think if our model is correct, based on our current fits, we guess it's something like 8 seconds. But again, I haven't shown you that full model, and we're still kind of working on it right now. But again, it's something we can't probe directly. It's fully from fitting this information.
Well, can you not? Well, you have this. Okay, presumably you have these process events occurring in rapid succession.
Yes. So if you look at, let's go back to some unwinding traces. So yes, you can have processive traces happen one after the other or with some break between them. I haven't analyzed the information between, on whether you have the time between processive unwinding, because it's hard to say if we're talking about an inherent state of XPD, when does XPD leave this state? Say, if we're looking at one of these processive bursts. So XPD unwinds the entire hairpin and begins and continues onto the opposite strand. Where did XPD change from being in a high processive state to a low processive state? Say it's a conformational state of XPD, it could be in that same conformation the entire time. Does that make sense?
Okay.
I mean, TJ's point is that you could look at the duration of the processive burst as an estimate of their lifetime, right? You have that data.
Yeah.
That's about, you have that mark somewhere, that's like 10 seconds or something.
For the duration of processive, so the average duration of a processive burst is about 30 seconds. But again, has XPD changed state in that time? We can't really say. But yes, we can measure that. I do have data looking at the likelihood of if burst n is processive, what's the likelihood of burst n+1 being processive? There wasn't a strong argument to be made with that, strong correlations that I saw.
So the number is exponentially decreases or something like that? no memory for
Yeah. No memory that I can tell.
But and then also the experiment where you try to form a complex and then move to the other side, that also puts a bound on how long that complex can live, right?
Right.
And so you said that was 10 seconds.
Yes.
And you said it was 8 seconds from the model.
Right.
So it's roughly consistent.
Right. And for burst duration, a non-processive burst, I think, is on average 10 seconds. Processive burst is on average 30 seconds. So if 10 seconds is long enough for XPD to change states, it could be changing multiple times during a single burst, especially a processive burst.
And also, can you argue that you need to have ATP, present to achieve this processive state, because you are incubating XPD and RPA separately for a long time, and a high concentration RPA. And if indeed this processive state can last like tens of seconds, as the data may indicate here, then you should be able to see them once you bring them back, right?
Yes. So I glossed over this for a physics audience, but I have in my Blink channel and in the XPD channel ATP gamma S at 500 micromolar concentration, which is quite high. For that reason, that perhaps in order to get good binding or to form this complex, XPD needs ATP. Could argue the merits over whether gamma S is good enough, but I did try to account for that. And yes, all of the data I've shown you has ATP gamma S mixed with XPD for loading and in the Blink channel during movement.
That includes the complex formation.
Including the complex formation data.
Okay, thank you.
So I have a more general question. So you spent quite a few years looking at this system and ruling out models and so on. What was the most surprising thing that you found to you?
The most surprising thing I found about
Just in general about this topic.
That's a good question. I think I was a bit surprised how much I like doing data analysis. I could make these plots all day. Yeah.
That's good enough.
Nothing scientifically that surprised you?
I was surprised by, well, every new iteration of the experiment that you do, you're like, yeah, making this change is the silver bullet that's gonna get me the data. Like, adding fret's gonna be it. Okay, fret didn't work, but this new analysis method, this is gonna be it. And somehow every time I was always surprised that it wasn't the answer.
All right, are there any more questions?
What about the public?
Yeah, so if I could follow up on TJ's idea. So with rep, for example, rep is in one state, in a translocation state on single-stranded DNA. So if it's bound to, for example, ATP gamma S, it would be in this translocation state. But then once it enters the ATP channel, it may be in an unwinding state. And so in the unwinding state, it may have a binding site or a stronger binding site to RPA. So I still think that even though you just did the comparison between binding to ATP gamma S and ATP, but I also think maybe there's like the translocation state and the unwinding state that may influence the complex formation.
Yeah, for those, I don't know that everyone could hear, but Monica asked, so her, one protein she's familiar with, rep, There's, has different interactions, whether it's in the presence of ATP, whether it's translocating or not. So she asked, maybe XPD has different states if it's moving along DNA versus being stationary. Since our incubations are XPD is stationary, perhaps that means it can't interact with RPA2 the same way it does when it's moving. That's possible. I don't really know how you would account for that exactly. I mean, what I guess I would hope is that you would find some ATP analog, non-hydrolyzable analog that produces the same effect, right? That, XPD is in this prepared state to interact with RPA. The translocating part, though, once we allow XPD to be translocating as it binds, we have the question of whether it's still a single XPD or not. So that's why we have this small binding site and why XPD is stationary during these incubations, because if we allow it to begin unwinding, a second XPD can just bind right behind it. So yeah, you'd have to change the experiment. It could be possible with some of more like what previous experiments in our lab with XPD, just mixed very low concentrations of XPD with ATP. So that was based on the idea that because the concentration of XPD is so low, if you see activity, it's likely to be only a single XPD. So perhaps RPA experiments like that could tell you something. But yeah, that'd be wild.
All right, if there are no questions, then we'll ask everyone to leave.
Turn this off.
Yeah.
Yeah, it's just issue to know viewers.
And stream.